Okay, so the first thing is to open the program. So you guys read in the tutorial here, as you read the first few pages, explaining what crystallography is and how crystals are grown and how we do the sitting drop method versus the hanging drop and so forth. And we would normally do this in lab if we had access to campus this semester, of setting up some crystals. Figure 2 here is actually some crystals grown by students that we used to take Polaroid pictures of in the past. Been doing this for quite a while. And then of course figures 4 and 5 are here if we can pretend to take these crystals to a X-ray source, a synchrotron source. I could fly you all to Switzerland and do that if I had an unlimited budget, but we don't have that. Or you can send them right off campus as we did at LSU. And you can take pictures of this with, not pictures, but you can take images with an X-ray beam. So you take an X-ray from a synchrotron, shoot it at your crystals, and the X-ray interacts with only one part of your molecule, which answers the question from our talks earlier of why it was difficult to get those structures for the copper enzyme. Is when the X-rays interact with the molecule, they interact only with electrons, nothing else. So when an X-ray, which is a photon of light, right, interacts with an electron, what does it do to the electron? Well, we have a couple options. If it's of the right amount of energy, you guys mentioned this in your talk earlier, it could promote it to a higher orbital, right? And then that electron could decay back to its lower orbital and you get fluorescence, right? You've seen that before, you've heard about that before. If the energy of the photon is of sufficient amount, right, like in these X-rays, right, it could take the electron and eject it from the molecule, right? That's what you were seeing earlier with the photosynthesis, photosystem 2, and with the copper enzyme. If it's not of the right energy to do those things, the X-ray can simply get diffracted, right? It just bends the X-ray and emits it off in a different direction, right? And that's what we're doing here. The X-ray hits the electron, right? It absorbs the energy and then re-emits it. And to simplify things, probably oversimplify it a little bit, when it absorbs the X-ray energy and when it re-emits it, do you think the electron is in the same place in its orbit? I know it's not a real orbit, it's a probability density, but do you think it's in the real exact same place? It's unlikely, right? But the time frame from when it absorbs it to it releases it will always be the same. So it releases it and the X-ray goes off in a different direction. That's all we're saying. Okay? And every electron in every molecule in every part of this crystal, which is compacted proteins, right? In this case, all compacted into a small crystal, are going to interact with that X-ray beam. So what you see is X-rays being spread all over the place. So we get a detector like you did at the dentist's office, right? You put the screen behind your tooth and your tooth absorbed the X-rays and you get a shadow where the X-rays didn't make it through. A very similar thing, we put a detector, we don't use film anymore, we use an electronic version now, but it's a screen behind this beam, right? That's what's shown in figure 4. As the X-rays hit the crystal, they're diffracted, and at some places on this screen, the X-rays come together and interact productively, right? They don't cancel each other out. And in some places, they're out of phase by 90 degrees, right? Or 80 degrees, or 70 degrees, or 60 degrees. So they get some sort of canceling of each other out, right? You don't see as dark a spot. So if you look at figure 4, you see there's a bunch of spots, right? That's where the X-rays interacted productively, right? And where you see no spot is where they canceled out. And those spots, those dark spots in figure 4, aren't just random. You notice they form patterns, right? There's some concentric rings. So off to the left here is a concentric ring, right? And then there's more around those, right? And this pattern of spots tells you something about where the electrons are in relation to each other. We won't get into the Fourier analysis of how you convert reciprocal space to real space and so forth. You don't have to do all that. But these spots tell me something about where the electrons are in real space. So the closer they're to space here, the farther apart the electrons are in real space. The farther apart they are here, the closer they are in real space. Okay, so you don't have to do all that math, but you can take that in a computer program and let it crunch on it for several hours or days, depending on the data set, and you get something like figure 5. Right? You get this cage network back. So what's shown in figure 5 is where the electrons were, right? So where the electrons might have been when the X-rays hit them. Okay? So there's so many electrons and so many molecules in this crystal that we pretty much catch the electron everywhere in its orbit or everywhere in its potential. So an ideal atom would look like a sphere, right? We don't see the nuclei. Why not? Because the X-rays don't interact with the nucleus. It only interacts with electrons. So all we see is a spherical ball. And if you look at figure 5, in the very center of the picture, there's a sphere of electron density, right? Everybody see that nice big sphere in the center? Whereas off to the side, you notice these aren't spheres anymore. They kind of look like they're spheres that are smushed together. They're merged, right? And that's because these two atoms over here are actually in a bond to each other, a covalent bond. So in one case here, look on the top right of the picture where my mouse is. You may or may not can see the mouse sharing my screen, but you'll see it in the video later. On the top right of the image, it looks like a bunch of spheres smushed together into a ring, right? There's 5 bumps on this ring, right? So this is a 5-membered ring. There's a similar 5-membered ring on the bottom left of figure 5. There's one on the bottom right of figure 5. There's also one on the kind of middle to top left, but it's turned 90 degrees to the others, okay? All those rings are part of a structure called a polyporphyrin ring, right? Much like the heme group in hemoglobin, right? And in fact, that's what this picture is, right? This is the heme group in hemoglobin. And those are the 4 rings of it. There's also a histidine ring that you can see kind of standing out on the bottom right that's coordinating the iron in the center. Right? And someone mentioned that earlier, that why were there so many histidines in certain things? Well, they're good at coordinating metals, right? Cysteine as well. Right? So what I want you to do is let's practice, or you guys can watch and do this along with me, but if you have questions, just ask. I'll show you what we're going to do. In the tutorial, I want you to run the DeepView program. So going back to our files, here in the program, we're going to click this thing. This is the only time you're going to double click something in the whole process, right? Is to open the program. We have some acknowledgement of who wrote the program. I'll skip that story on how it was created for time. And I'll put this box in the top left of my screen, right? And not make it go all the way across. I'm going to need this side box for something later. Okay? So next thing, I've run the program. I want you to open the file tutorial.pdb. And there's a certain way you should open this. You don't double click it. Okay, so I don't need this folder anymore. So in my program itself over here, I'm going to go to File. And there's lots of things I can do, lots of things I can open. And I was asked to open the tutorial.pdb file. So open PDB file is the first option. It'll open a navigation window. And go find in your folder I asked you to put on your desktop from all the things you downloaded, go find tutorial.pdb, right? Like that. Don't double click anything, just hit open. If you do it correctly, you'll get this general communication or warning box. You don't have to understand what it's telling you, it's just there's some atoms that aren't connected as they should be, and just click OK and it should open the whole thing. Right? I'm going to close all these extraneous windows. This is the only thing I need open. Right? And I'm going to set it to a size that just fits on my screen.
Wait, can you go back and which file do we...
Say that again?
Which file to open?
So we're opening the program itself, which is this one with the little icon, the Swiss PDB Viewer program. When you double click that, it'll open this window up top. Then in the tutorial, I'm asking you to, once it's open, run or open tutorial.pdb, and I'll close it and do it again. Right, so I'm going to open the program by double clicking this, close the acknowledgements window, put this box in the top left corner of your screen, make it cover about 3/4 of your screen. Then go to File, Open PDB File, and I'm in the folder where I downloaded all those files from D2L, right? The ones I gave you, right? All these files you should have downloaded. I have some in here that you don't have, but you should have downloaded all of these. And click on Tutorial, and then click Open. You should get this warning box. Click OK, and it should open this. You can close this little window, right? And I want to scale this window, I can scale it any size I want, to match the one up top and fill most of my screen, right? And if I move it, it should move with the top window. Right, so I'm filling up most of my screen, so we get a nice big view. Leave some space on the right over here. We'll need it in a little while. Right, so back to the tutorial. I've got it open, right? And this is the solution structure for Lysozyme, right? Lysozyme is an enzyme that prevents bacteria from repairing their cell wall, basically. So you don't have to work very fast if you're Lysozyme. You punch a hole in the submarine's gone. So it just punches holes in bacterial cell walls faster than they can repair them, and that kills the bacteria. You produce lysozyme all the time. It's on all of your liquid secretions from your body, right? That's mostly what's in those little crystallized tears you have every morning in your eye, right? It's mostly crystallized lysozyme. So it's very easy to crystallize, which is why it was the first one done. Okay, so on the program itself, you notice there's these 3 buttons. It's a little hand button, a pair of windows, and then this little round rotational thing, arrow thing. All right, we're going to learn what those 3 things do. And they're your basic utility tools. So going to the program, I have the hand tool selected. I can select any one I want. So I'm going to select the hand tool, put the mouse in the field out here. It should show you the hand tool. And using the left mouse button, if I click and hold down and move it, it moves the molecule around. This is called the translate tool, right? I can move it left, right, up, or down, right? Any way I want. Right? If I click the second one, the window tool, and I do the same thing, I put the mouse in the field out here and hold down the left mouse button and move it, it should zoom in or out, right? Either way you want to move it. So I have a translate tool and a zoom tool. Lastly, with the arrow, if I put it in the field and click and move the mouse, it'll rotate it in the direction I'm moving. Okay. Now, when we did this back in early 2000s, and all students do this, if you take the mouse and do something like that, it would crash your laptop. Right? But modern computers can handle this rather well. Right? So practice using these functions. I can rotate, I can zoom in, move, say, molecule left or right, and you should notice that I found a tryptophan residue right here in the center of the screen. So I'd like to center on that tryptophan, get it in my view. So I'm going to zoom in a little more. Right? You notice it is 3D. So I rotate it a little. Right. Somebody's mic is open. You can rotate it a little bit, and then I can translate using the hand over to here to get it in the center again. We'll try zooming one more time. And if I rotate it, it may go off the screen. Right? It's not doing a bad job of staying, but if I rotate over here, it goes off the screen. Right? I'd like to keep it here. Right? So I can zoom out some, right? Rotate and translate. How am I doing all 3 of those things without changing the function over here? You notice I kept rotate selected the whole time. I can rotate, I can also zoom out or in, and I can translate without selecting the other functions. What I'm doing is using both buttons on the mouse. So you need a 2-button mouse to do this, right? And then the way of doing that on a Mac does not work with this program. So if you have rotate selected and you left click, it obviously rotates. But if I use the right mouse button, it translates. And to zoom, I use both mouse buttons at the same time. Okay? The scroll wheel does nothing. Okay? So left click, rotate, right click, translate, and both buttons zoom. That way you don't have to worry about changing all of these. Everything's literally at your fingertips. You can do all 3 functions. Okay? So we got familiar with those. Let's go in and find a tryptophan and get used to using these functions. So searching this molecule, there's quite a few tryptophans in it, but I'm going to use the one that's dead center right here. Right? Tryptophans are pretty easy to find. Right? If you don't know your amino acids yet, you can scroll to the end of the tutorial and you'll find the structures there. There's pictures for you. Okay, so back to the tutorial. I'm going to rotate, zoom in on the tryptophan. I may have to move it down a little to keep it in my screen. Right, so I get a nice clear view, relatively clear view of the tryptophan. Okay, so practice doing that with a few amino acids. There's another tryptophan over here. Pretty easy to find. Right. This one up here on the top left would be what amino acid? The one I'll put it at the very top of the screen. What amino acid is that? Anybody have a guess?
Is that proline up there?
That's proline, exactly. So I'm going to move down, zoom in on it, move down some more. And if I rotate it, may go off the screen, you see, because my center of rotation is the center of the molecule right now. But yes, this is proline. So this is the amino nitrogen, the alpha carbon, and this is the carbonyl. You'll also notice that this is shown as a carbon to oxygen bond, not a double bond. So no double bonds are shown as double. They're just connections. That's all these lines are. Right? All the lines are bonds, but they're showing connections, not necessarily double or triple bonds. But this is definitely proline because there's the propyl group that wraps around and binds to its own nitrogen. Okay. Right, so what if we wanted to see that tryptophan again, but I did something crazy like this. I'm completely lost. I don't know how to get out of this molecule, and for some reason I can't zoom out. It won't work. Completely lost. There is a get out of jail free button if you think about it. So over here on the top left, click the button. There's the hand tool, but to the left of it is this very left button. Click it and it resets the whole molecule. So just in case you get lost, you can always reset it and find your way out of there. Okay. Right, I want to cover a few more functions, just 3 more for now. We're not going to do everything this program can do, but I want you to get the basics down. So over here, the next 3 functions I want to cover are number 5, number 8, and number 10. Okay, so on this little diagram over here. So in our program, that's the one that has a red dot with a line between it, and then there's this 1.5 angstrom measurement above it. So what do you think this function does? It's got a dot with a line drawn between them, and then some kind of 1.5 angstrom text above it.
Hydrogen bonds?
It could measure hydrogen bonds, but the idea is it just measures distances, right? So I'll show you how to use it in here. Let me go find a tryptophan again, since we're familiar with those. I'm going to use this one right here since it's kind of all by itself. Right, so that tryptophan right there. Everybody see its 2 rings? It's kind of at an angle to us right now, but you can see it's 2 rings. Right, we'll get a better view of one in a minute. But this part, as you know, in tryptophan, this is a benzene ring, right? It's an aromatic ring. I'd like to find out how far is it across this ring. Pick any 2 atoms, right? So I'm going to select this function. It's the distance picking function. And underneath it in red, it gives me some instructions. It says pick 1 atom. Okay? So I'm out here clicking all over the place and nothing's happening because I'm not clicking on an atom. Atoms occur at the intersections of lines. But how do I know if I've clicked on it? I really have no indication in the picture down here. But as I'm about to click on it, right here it tells me if I hover over something, it tells me when I'm about to click on it, right? So if I click here, this is the thing I'm about to assign to that point. But I'm clicking and nothing's happening. How do I know if I've done it? Well, I want to save you the trouble of the computer program laughing at you. Let it laugh at me here for a moment because you don't want this to happen to you. If you rapidly click here twice, it says, yes, genius, those 2 atoms are 0 angstroms from each other because you clicked the same thing twice. Okay, so the computer's laughing at me for a moment. I want to try again. This time when I click it once and it says pick first atom here, right? How do I know if I've clicked it? Now the construction says pick second atom. Oh, so I know I've clicked it. So now I'll go click on this one. How do I know if I've clicked this one? It'll actually draw the line. And it'll put a distance measurement there. Okay? So the distance across this benzene ring is 2.74 angstroms, right? Which is on par with a hydrogen bond, yes, but this is not a hydrogen bond. It's the distance across this ring. Okay? And if you measure it across the other way, click here. How do I know if I clicked it? Pick second atom. Click here. Right? It's kind of hard to see the measurement, so I'm going to hit rotate here, turn it. Might still be hard to see because they're right on top of each other. Let me do the 5-membered ring for you so you can see a separate measurement. All right, so again, click the distancing function, click this one, across the ring, second atom, 2.24 angstroms. How do I get out of this function? I can either go click rotate like I just did, or just hit the escape key, and it gets out of that function, and you're back to normal. Okay, so clearly across the 5-membered ring here, what I'm considering across, because I can't click the middle of a bond, is shorter than the 6-membered ring, but you already knew that. It's smaller. Okay? How far is it from this atom here, this carbon, to this carbon? Right? How long is this carbon to carbon bond? Well, which carbon is that? Well, here's the backbone of the protein over here. So this is the amino group, right? The carbonyl carbon, or so the alpha carbon, and the carbonyl carbon over here. So this is the beta to gamma carbon length of tryptophan. How far is it? Well, how long is a typical carbon-carbon bond? Somewhere around 1.4 to 1.5 angstroms, right? So I'm going to click here, click second atom, and it's 1.52 angstroms, right on par with what you'd expect for a carbon-carbon bond. If I check the alpha-beta length, right, from here to here, shows a similar result. Right, so this is your typical carbon-carbon bond length. Right, you can do that between any two bonds in here. Right? If you wanted to measure hydrogen bonds, like someone mentioned, I need to find two electronegative atoms that might form a hydrogen bond. Let's try to find one real quick so you can see what's going on. Perhaps these two. I don't know because I haven't checked it. Let's find out. So do you see this backbone carbonyl sticking out here? Right? Not a really good view of it till I teach you how to do that other program component in a second. There's a backbone carbonyl, and this is the end of an arginine, right? Arginine side chain there, right? So this is that guanidinium group at the end. Is this oxygen close enough to this nitrogen to form a hydrogen bond? Let's find out. So I click here, click on this oxygen, pick second atom, click here, and it's 3.15 angstroms away. That's entirely close enough to be a hydrogen bond. Hydrogen bonds are roughly 2.5 to 3.5 angstroms, so that could be a hydrogen bond, assuming this nitrogen has a hydrogen bond. Right? So you can check all those things. Right? So that's one of your functions I'd like you to learn. The other is this number 8 in the example here. It says a dot with a question mark next to it, and then there's this Lu41 written above it. Right? And the Lu41 is just a label. So this is a labeling function. So going back to our tryptophan one more time. So there it is again. If I could click this labeling function, then hover over an atom, how about that one there? Right? It'll tell me what it is right here. It's tryptophan 63. That's the residue number in the sequence. It's CB and then a bunch of numbers. So what does this mean? If I click on it and label it, get out of that function, it tells me it's tryptophan 63 CB, and the other numbers were just its 3D coordinates in this program, right? So what does this mean? It means it's part of an amino acid, tryptophan. It's the 63rd amino acid in this protein. This is the element, carbon, and this is the position, beta, right? We said this was the alpha carbon, this is the beta carbon. So if I try to label this one, what should it say? Right, I can hover over it to find out, and it says Trp63Ca, which makes sense because it's the alpha carbon. There it is. If I rotate and do the next one, this position here should be the gamma carbon, right? So if I click on it, it should read Trp63 carbon gamma. That's exactly what it says. Carbon gamma. Right? So you can label anything you want in the structure. Any atom you can click on. Okay? You may notice something unusual in the structure. It doesn't look like the rest of it. Anything in here stand out to you that doesn't fit with the rest. Clearly all the carbons are in this off-white backbone. Nitrogens are blue. Oxygens are red. Sulfurs are colored yellow. If we had any phosphates in here, they'd be orange, but there's no phosphates. This is not a nucleic acid. Right? So we've got a couple disulfide bonds here and here. But what is this? What is that thing? It's not any connections to anything else. So if I were to ask you to draw just an atom without any connections, it would just be an infinitesimal dot. You wouldn't be able to see it. So when it's a single atom all by itself, the program draws it as a sphere. And we generally do these for metal ions in the structure. So if I go and label this thing, it'll tell me what it is, right? So this is its location, just an assignment, but this is the element. What is it? It says Cl. So just this sitting there, it's not your normal metal. What is it? Cl. We can't type in lowercase in this program, unfortunately, so it's a chlorine atom. Okay. If there's any others in here, we should see those as well, but this is just a chlorine that's just sitting out there in the structure. Okay. The last thing I want you to learn is number 10 here, right? And that's this function right here, right? So if I want to pay attention to this chlorine atom, you notice I had trouble getting it on the screen and getting it to stay when I was trying to zoom in, right? I really want to take a look at it and see what its neighbors are, but it keeps bumping all over the screen. I'd like it to stay still. So I click this centering function, which is an eye over 4 arrows pointing to a dot. Click on the chlorine atom, and it centers it in my view. Now, the program assumes you only want to center once, so it put it right back to rotate. And now, no matter what I do, that chlorine doesn't move, right? I can move it over here, but it's still my center of rotation. Everything's revolving around it when I move, right? I can do the same thing with, say, the tryptophan, and the tryptophan won't move, right? That atom I clicked on will not move. Everything rotates around it. So I can get a nice view of things if I want to keep it in my screen. And so if you learn your distance, labeling, and centering functions, then you can do all those things as well as manipulate the molecule with your mouse. And so that's all your basic functions right there. Right? Do you guys have any questions on those first before we move on to the next part?
Is there any of those you have concern of USCF Chimera being unable to do so?
I think the Chimera program has similar functions, but I'm sure the buttons look a lot different.
Because so far everything that I saw we've done in lecture or a project. So if program is able to do them, is it a reasonable substitute?
Sure.
Sure,
if it teaches you the same idea, it's fine. But make sure that it can also open the next file I'm about to give you. So this is the meat of the exercise coming up. These were just basics of getting familiar with the program. Okay, so try doing this on your program as well. So we're going to move on to number 4 and open an OMAP. Okay, so this is the file that was generated, much like figure 5 up here, from all that processing of X-ray data. Okay, so the way you open that is I'm going to reset this molecule real quick. You don't have to do that, but I just put it back in the center. I'm going to go to File and Open Electron Density Map. Okay, I'm not going to open a PDB this time. We already have it open. We're going to open the electron density map that goes with it. Okay, so go to Electron Density Map, scroll over to DN6, because that's the type of map it is. On a Mac, it'll populate this right away. On PCs, it doesn't understand what an O map is. It's just looking for regular DN6 maps. So you got to tell it to look for all files. And now you can go down and find tutorial.omap. Highlight it and click Open. Okay. It's going to give you a warning that one of the parameters is not where it should be, and that's okay. Just click OK, and this box should pop up. Right. We want to make some changes here in this box. The default program, I think, has 7.5 written here, which is large and probably a little too cluttered to see what's going on. I like to change it a little smaller, but not too small. What we're doing here is telling it how far to render the density in our screen. In other words, how much to display. So I want to make a cube of density that's 5 by 5 by 5 angstroms. So make sure this little radio button's checked. 5 by 5 by 5 angstroms. Contour a 1 sigma map, right? Make sure it's a dotted map. Don't check the other one, just leave the 1 sigma. Then click a color, right? I like this bright green color. I think it shows up well on this screen. We'll see how well it shows up for you. You can choose a fuchsia, a pink, or something like that. I would stick with greens and pinks or yellows or something like that. Stay away from blues and reds, because then you won't be able to see your nitrogens and oxygens respectively with this on top of them. So green works really well for this. All right, so I'll click OK, and it should show the map. Okay. Most likely, yours, if you're first time running it, looks like this, where as you turn it, it disappears, which is sort of useless as you want to see how it turns with the structure. So the way to do that is to go under Preferences, it says Prefs at the top, into Real-Time Display, and make sure electron density is checked. Okay, so under Preferences, Real-Time Display, make sure electron density is checked. Then when you turn it, it should stay on the screen. Right? It is more computationally intensive, but for modern computers, this isn't a problem. In early 2000s, this was an issue. Okay? So now if
Wait, what was the last
Go ahead.
thing that was checked under display?
So under Preferences, Real-Time Display, make sure the electron density is checked. The others are on by default, but electron density is off by default. So go in and check it. Make sure it's got a check box somewhere.
Thank you.
Then when you rotate, it should stay on your screen. It shows it in real time. It is more computationally intensive, but it should be fine. Okay. So now let's go find our tryptophan again. Right. I'm going to pick a different tryptophan this time to show you how I can move over there. So let's find another one. How about this one right there? All right, so I'm going to zoom in on it. And there's another tryptophan. You notice, as I'm trying to get a picture of it here, the density stayed over here on these other atoms. The density will always be around the atom you center on. So if I go and use my centering function on, say, this atom of tryptophan, the density will follow it over here. Right? If I rotate again, do you see that the electron density looks like it's been sort of shrink wrapped onto the structure? I know it's difficult to see if you're not the one turning it. I get that. But I'm trying to get you a clear view to show you that even edge on, you see that this electron density looks like it's been like shrink wrapped onto the atoms. And that makes sense because the electron density is what they use to figure out where these atoms were. Right? And tryptophan's 2 rings, as you see here, are truly planar. Right? You see the density looks nice and flat. Right? You can even see the hole in the middle of the benzene ring from the density. There is no physical space in there, but as far as we rendered the density, it exhibits a hole. Right? The 5-membered ring is not quite big enough to generate a hole by this resolution, but you should see a dimple where it goes in. It's a torus. Right there. Okay, so you've learned how to open the file. You've learned how to manipulate the mouse, manipulate the screen, center, measuring distances, and labeling. Okay, the last thing I want you to learn how to do before we start practicing is how to change the amino acids. Okay, so I'm going to say for a second, let's go find another molecule. I don't want to use tryptophan this time. Let's go over here and use, how about that proline you guys were looking at earlier? Right, so this guy over here. Right, so I'm going to zoom in, try to get proline in my screen, center on the proline. I'll use its alpha carbon, right? And the density will follow me over here, right? And does this density fit the proline? Right, well, what do I mean by does it fit? Right, we got to make up some rules here. So when I say it fits, I mean does it shrink wrap well? In other words, 2 things have to be true. I don't see lines, bonds, and atoms sticking out of density, which would indicate there's an atom here with no real density in the real world to support it. Why did you assign it there? Right? I also should not see any empty cage. Right? There shouldn't be a blob of density here without an atom assigned to it, because there should have been one. There's real density for it there. So what we see here is all the atoms fit and all the bonds fit within the density, and there's no empty density anywhere. And some of you, really eagle eye, are pointing out, like, what is this big blob sitting out here then? And you're absolutely right. There's a blob here with no atoms assigned to it. We'll get back to what that guy is in a second. Right? But you're also pointing out there's density over here on the bottom left of the screen and nothing supporting it. Well, what you're seeing there is, remember, this thing occurs in a crystal. So this protein repeats over and over and over. And you're seeing the next contact to the next copy of this protein over here. Right? So it doesn't draw the protein numerous times. You can tell it to, but once is enough. So this is just the next protein in the long chain of proteins in the crystal. Okay, so this proline actually fits really well into the density. You can even see the little bumps from each of the atoms, right? Which is sort of spectacular on the edge here, right? The closer you get to the surface of the protein, the less likely they are to fit so well, right? If you go deep in the interior, they generally fit much better because they don't move around as much from copy to copy in the crystal. So if I go back to this other tryptophan, you notice how well it fits, right? Fits perfectly well in this tryptophan, right? So I want to do one more with you. Let's find a different one this time. How about this amino acid? I won't tell you what it is right away. I want you to guess. What do you think if I can get it in the screen. This one is. Let me center on it for you real quick. Everybody see this one here? It's got a 6-membered ring in it, and then it's got an oxygen sticking off the end. And this is where it is. What do you think this is?
Is it tyrosine?
That is tyrosine, exactly. So if I center on it, I'm going to center over here on the alpha carbon again. Zoom out just a little, right? You see it's definitely tyrosine. I got it sticking off to the left there. Right, so here's your alpha carbon, beta carbon, gamma carbon. There's 2 delta carbons because it splits into the ring, right? 2 epsilon carbons, right? And then it converges onto a single carbon with the OH group on here, right? So it's definitely tyrosine. And my question is it really tyrosine? How do I know? Based on the density, could it be something else? So I'm going to play devil's advocate for a minute because obviously it's tyrosine. I think it might be histidine. Okay? I'm clearly wrong, right? But let's pretend and convince ourselves that it's tyrosine and not histidine. Because histidine also has a ring like this, right? It's not a benzene ring, but it has a ring. So what I'm going to do is use these last 2 functions in the program up here, these mutate and torsion functions, mainly the mutate one, right? So I'm going to zoom in, get a good view of my beta carbon here, right? So alpha carbon here, beta carbon here, right? And then there's the ring. It's a good view of it right there. Nothing in my foreground or background, and I can click on it. I click on mutate once, it says pick one group. And I can click any atom in this tryptophan, or sorry, in this tyrosine, and it'll change it. So after all these years of using this program, this is still the one part, which I don't know why it uses the JavaScript for this part, if it is JavaScript. But it's the one part where it still might crash on you. Right? So when I click on this, it's going to give me some options of what to change the amino acid to. I need to choose it quickly or it generally crashes the program. It's probably a memory leak somewhere. Right? But if I click on this and I said, I think it might be histidine. So I'm going to click this and quickly find histidine. They're in alphabetical order. I don't want to leave it on the screen too long. If you're watching this on the video, pause it and you'll see that box that pops up. But I clicked histidine, right? Now I want to come up here and click mutate, and it says accept my mutation, tyrosine to histidine. If you click the wrong one, just click no and try again. Click mutate, click here, go to histidine, then click mutate. Yeah, I clicked the right one, hit yes. And now go back to rotate, and histidine has been placed there. Okay, does it fit better than the tyrosine? Well, this clearly looks like a 6-membered ring, and I put a 5-membered ring in it. You can see all 6 bumps, right? So obviously it's not histidine, but I'm still not convinced. It looks like it fits. If only I could turn it a little bit, it might fit. Just playing devil's advocate here. Click on torsion, click on any atom in the now histidine molecule, right? And some arrows will pop up. Do not alter the arrows underneath the torsion. Okay, those change the phi and psi angles of the backbone, and we won't be changing that. They're going to be correct. The 2 on the side means that histidine has 2 bonds that can rotate. If it had more, there'd be more arrows here. So histidine has this bond that can rotate. Right, and the faster your computer, the faster this goes. So I'm trying to go really slow with it. And of course I can rotate the ring. Okay. So I did some rotations, perhaps I changed it, hit torsion. Does it seem to fit better now? No, I clearly made things worse, right? It doesn't even fit remotely close to this. So I want you to do this, and I'm not going to grade you on do you get it exactly to fit properly. That's not the idea. The idea here is you convince yourself what amino acid belongs here. So I'm going to go to mutate, and clearly histidine I can't get to fit. So I'm going to go put it back to tyrosine. Right? Click mutate, accept that mutation. Does tyrosine fit? Well, it looks like it fits, except you notice the angle's off a little bit. Right? It's not quite the right angle. So I'm going to put it here. Hopefully I can click on it and change the angle of that ring and move it just a little so I can get it to fit. Now does it look like tyrosine fits better? Yeah, I got it almost exactly. I'm not going to grade you on if you get it exactly or not. The program here is designed to help you learn your amino acids. So once I get it and go, I'm convinced that is tyrosine. Okay? Because I've centered where I can see the density for this OH group. What if you couldn't see the density out here? What if I had centered a little farther away, perhaps like this? Now can you tell it's, move it over so you can see it. Can you tell if this is tyrosine or not? You can't see the end because you told it to only draw it so far away. So I can't tell if this is tyrosine or phenylalanine. The only way to tell is to change your view, move farther out so I can see all of it. Now can you tell it's definitely tyrosine? I see the bump for the oxygen. Right? If this didn't exist and there was just a little bump here that did like that, it would be phenylalanine. So all I need you to do is convince yourself of what the amino acid is. Okay? So that's what you're going to be doing by selecting residues and corresponding densities. Okay? So I want you to practice this for a minute. Right? So if you're doing this with me, or if you're doing this on your own, you can pause the video and watch it later and pick up. And if you're watching this later, I want you to pause it and try doing this. There's 20 amino acids in the structure. This took a while to find back in, I don't remember when I did this, early 2000s, late 90s, somewhere around there. Right? So there's all 20 amino acids are represented in the structure. Right? It's easy to find today with the hundreds of thousands of structures deposited, but back then it was not. And so I want you to find every amino acid in the structure. I want you to do it one of two ways. One way, do it like this. Go in, zoom in on any one you want, and say, what is that? I'm just going to pick one. What is that amino acid? Right? Based on what I see, I think it is alpha carbon, beta carbon, and then a benzene ring. That looks like phenylalanine. So I'm going to label it. I'm right, it's phenylalanine. Right? If you label it incorrectly, guess again. Right? Doesn't make any difference. So I want you to do 10 of them that way. Find 10 unique amino acids that way. Happens to be another phenylalanine right behind it. Right? So find 10 that way. Here's an arginine over here. Right? Here's a glutamine there. Here's another arginine down here. You found this proline for me already once. So go through and find 10 that way. And then once you've checked off 10 on your list, right? For the other 10, I want you to do it the other way. Let's say I've checked off 10 of these on the list. I have yet to find a, I don't know, let's pick lysine. Right? So I look here, lysine. Now I have to go find a lysine. Instead of looking at the structure and naming it, I want you to pick a name, lysine. Let's go find a lysine. So we start scanning through the structure until I find a lysine. There's one there. You'll get better at it the more you do it. Right? Of course, you have to know your amino acids. So here's a lysine down here. Right? How do I know? I will label it. It's definitely a lysine. Okay, so do 10 of them where you just look at the structure, right, and label what it is by guessing. And then of the 10 you haven't found yet, go back and say, which ones haven't I found? I haven't found a cysteine yet. Well, there's one right there. Right? And then go label it and see if you got it right. So do 10 one way and 10 the other day. And you can pause the video to finish those. While you're doing those, I have two other questions in this tutorial for you. Right? Also, you'll notice in here that, well, not this structure, we'll get to it, that you should find some ions or isolated atoms like this one right here. Okay? Right? We already labeled this one as chlorine. Right? So do go through and find whatever amino acid you want to practice. And when you think you're ready, and kind of like a little mini test for yourself, I want you to open this other file and do this. So let's show you how to close and open files real quick. I want you to file, discard the electron density map. And you notice it will go away. It kept the PDB file. I just discarded, file, discard, and of course I already got rid of it. There's nothing to click, density map. Then I want to go to file and close, and it'll close the current electron structure, or current PDB file, and it should go away. If you go to file and quit, it'll close the program. Okay, so file, discard electron density map, and then close that layer. So on the tutorial here, I want you to now open 1YOJ. So go to file, open, PDB file, 1YOJ. There it is. Open. Now this one asks you to load some models, and you can do this on your own. If you want to load all 20, it'll look like a snowflake, right? This huge snowball. But it's done by NMR spectroscopy instead of X-ray. So we're only going to load one model, right? We get a view of it. Something new here that you haven't seen before in the other structure. There's a new color, right? So if I pick one of these amino acids, for instance, how about a lysine again? Right, so here's a lysine sticking off here. You notice each of these carbons has two other little light blue lines coming off of it, and the nitrogen at the end has three of them. Right, so these little light blue things represent hydrogens. We could not see them in the X-ray structure, and why is that? Well, how did we get the structure through X-ray crystallography? We shot an X-ray beam at it. The X-rays interacted only with electrons, not the nuclei. And if you have a hydrogen attached, where does the electron, the pair of electrons in that covalent bond, spend the vast amount of time, or have the highest probability? Not around the proton, right? Around the other element. So you don't see them in X-ray crystallography. However, with NMR, we're actually seeing nuclei, thus the N in NMR. Right? We don't see electrons here, we see the nuclei. So in this case, I can see all the hydrogens because they do have a proton. Right? But it's really not that helpful because you know where they are. Right? You know where all the hydrogens are. So they're kind of just cluttering up our image. So if we go into display, we can uncheck show hydrogens. And now it looks more like the structure we're familiar with. Right? There's our lysine again, the way you're used to seeing it. Okay, so in this one, this is the one I want you to pause and do. Find all 20 amino acids, 10 one way, 10 the other way, and then answer one more question for me. In this structure, there are two metal ions, right? You probably can pick them out of the picture already. I want you to tell me what element each one is and how far apart are they in angstroms. So go ahead and pause the video and try to get all 20 of those labeled 10 one way, 10 the other. Figure out what element these two are and then how far apart they are. Okay, so I'll do a few of these. So this one, if you've completed those, let's try to guess what this amino acid is here. Anybody have a guess? We've done this one already.
Which one are we looking at?
Excuse me?
Which one are we looking at?
Oh, so you can't see my mouse. I'm sorry. Let me put it at the very top of the screen for you, in the center. Top center.
Oh, tyrosine?
Tyrosine. Sorry, I forgot you can't see my mouse on this thing. That's exactly right. So we'll label it as tyrosine. You're correct. What about the one on the left here that has something yellow on it? I'll put it in the center of the screen again, at the top. That one. What might that be? Maybe I can move it off to the... No. I'll do this. Let me center on it for you so you can get a better view. Very center of the screen, the one that's not moving. What do you think that is? Here's the alpha carbon, right? And then there's a CH2 and then something in yellow. And yellow represented sulfur. So what is this guy?
This.
Cysteine, yes. So if I label it, I get cysteine. Right? Sometimes you can mistake it for methionine, but remember methionine also has another carbon at the end of it, and if you look just to the right of that, there's methionine. Right? You notice there's an extra carbon right between the alpha carbon and the sulfur, and there's also an extra methyl group at the end of methionine, thus its name methionine. Okay, so we'll do 10 of them that way, find them, and then there's some others you can go find, 10 the other way, and hopefully you find all 20 amino acids. They're all represented here somewhere at least once, some more than once. And lastly, you were asked to find out what are these two metals. Right, so we'll label them. So this one is a zinc atom, and this one is also a zinc atom. The numbers here, 37 and 244, are basically their ID numbers in this structure. Everybody gets an ID number, like the amino acids are assigned from 1 to whatever in their sequence. So it's just zinc number 37 and zinc 244. They're still zincs, given by the element afterwards. And finally, you were asked how far apart they are. So you click the distance measuring function. Hover over the first one, make sure you're about to click on zinc. So hover over it, click it. How do you know if you've clicked it? It says pick second atom. Then go over to the other one, and it tells you they're 13.21 angstroms apart. Like that. Okay, so that's our tutorial on getting you used to the amino acids. It's okay if you have to look at this to figure out the ones you haven't found. You'll get better at it as you memorize those structures. Okay, so let's close this one real quick. So we'll go to File, and we notice we didn't open any electron density because this had no electron density. This was done by NMR. So we just go to Close. The next series of these, I just want to show you a few of them. You don't actually have to open them with me. You can just play along. So we're going to open 1KYC, JY4, JYB, and so forth until we get to the end of these practice problems. Okay, so we'll go from 1KYC. This is going to be a structure of an alpha helix. Okay, so it doesn't really look like an alpha helix at first glance, but if you make it a little easier to see, it will. Okay, so I purposely didn't show you this window for the last problem. So now I'll bring up a window that would make the last problem trivial. We're going to go up here to Window and open the control panel. That's why we left room here on the right. Okay, so the control panel here lists the amino acids in order, which would have made the last search and find function trivial, but I want you to learn them first. So these show them in order, and I also can show their side chains and backbones here. So if I click the word side, it turns off all the side chains. Of course, I can always put them back on like that. And if I click it and turn them all off, now can you see that this is an alpha helix, just looking at the backbone? Okay. And in an alpha helix, what holds it together? The R groups were not involved. You notice they're all kind of pointing outward, not doing anything here. Right, so what's holding this alpha helical shape? I'm sure you guys had this in your lecture course. Anyone remember?
Is it the interaction backbone?
The backbones are interacting with each other. In what manner? What's the thing holding it together? Sorry, I couldn't hear you.
Alpha helix?
It is an alpha helix. What holds it together? What interactions among the backbones?
Hydrogen bonding between the backbone carbonyls and the amino groups.
Yes, so the backbone nitrogens and the backbone carbonyls are hydrogen bonding. So let's pick a pair here and see how that's happening. So can you see that this carbonyl right here in the center of the screen is pointing almost directly at this nitrogen to the left, right? It's not exact, but it looks like it's pointing right at it, right? How far apart are these? Well, you know the nitrogen has a hydrogen on it. We can't see it because this was done by X-ray crystallography. But how far apart are they? So if I measure the distance from this nitrogen to the oxygen of the carbonyl, it's definitely in hydrogen bonding length, right? It's between 2.5 and 3.5 angstroms. So 2.86 is an ideal hydrogen bond, right? You notice that's happening elsewhere. This one is also doing it from this nitrogen to that carbonyl, from this nitrogen to that carbonyl, right? They're all in hydrogen bonding length. And this is happening all along the structure as you go around, right? So all along the thing, it's being held together by hydrogen bonds. And the hydrogen bonds of which one is bonded to which other one, we have a formula for it. So if I were to label this carbonyl, right, it's isoleucine number 7, it's oxygen atom. And if I were to label this nitrogen, it's glutamate 11's nitrogen atom, right? And the formula is the carbonyl of amino acid N is hydrogen bonding to the amino group of amino acid N plus 4. So you add 4 to 7, you get 11, and that's the two that are hydrogen bonding. Right? If you had to guess, this isoleucine, its amino group, which is back here, remember this isoleucine's R group is there. Right? We turned it off. So this is the alpha carbon. This is the amino group, right? If I label it, you'll see that. The amino group of isoleucine 7 is hydrogen bonding with the carbonyl of what amino acid? Now, you don't know the name of the amino acid. I turned off the R groups, but you should know its number. So this carbonyl up here should be number 7 minus 4 would give you 3, right? It's leucine 3. So this again is another hydrogen bond. So every amino acid in here is making at least one, most of them 2, hydrogen bonds. Right? That's what holds this whole structure together. Now if I were to take it and turn it on end, so you're looking down the barrel of this thing, there is no hole here, we're only showing a stick figure. But I'm taking it and turning it on end so you're looking down the barrel. Then I'm going to turn on the R groups one at a time. Okay, so I'm going to turn on the glutamate, there it is, right, sticking out. I'm going to put it so it's at the very top, pointing up. So glutamate is pointed almost straight up. And the next one, when I turn it on, you notice it's pointed this way. It's another glutamate, right? How far is the angle between this one and this one? Well, we did this, I don't know if you did this in lecture, we did it in the lab for sure. The angle between them is determined by how fast they rotate around this helix, right? So if it takes 3.6 amino acids to make one full rotation, right? How far is each one rotating? Well, it's 360 degrees in a full rotation, and it takes 3.6 amino acids to go all the way around. Each one's turning 100 degrees. So the angle between this one, the glutamate number 1, and that's this one up here, and glutamate number 2 is 100 degrees. And if I turn on leucine number 3, it should be yet another 100 degrees from this one. Arginine 4, another 100 degrees, right? So if I go to arginine 5, where it should be almost lined up with the glutamate number 1 again, except we've turned 400 degrees, not 360. So you see it's a little past that point, right? And if I keep going around, putting these on, you notice they just keep rotating 100 degrees each as I go around, all the way to the end. And that's how the R groups stick out from an amino acid. We can also show that with our ribbon drawing in here. It's a very crude ribbon drawing, but you see they all stick out from the ribbon. All the R groups are pointing outward radially. Okay, we can also show it as a space filling model, which can be useful for looking at interactions. Okay. All right, so that's our alpha helix. We're going to do the same thing with a beta sheet. So 1JY4, so I'm going to go to File and close this one and open 1JY4. Right, this was again done by NMR. So we load one model. Right, and again we show all those hydrogens, so we can go up to display and turn off the hydrogens. And it may not immediately look like a beta sheet, but let's get rid of those R groups and take a look at the backbone. Right, now does it look like a beta sheet? And you're probably used to seeing beta strands running next to each other like this, but beta sheets also have this propeller twist to them. If you look at it from end, you can see there's a propeller twist to it. Right, they're not perfectly flat. Right, now is this strand or this protein a parallel beta strands, or are they anti-parallel, or is it mixed? How do you figure that out? So I first need to figure out which direction the strand's going. So let's just pick the strand in the center, right? The one in the very center here, this one, and see which direction it's going. So I find nitrogen, carbonyl, alpha carbon. That's backwards. So nitrogen, alpha carbon, carbonyl. Nitrogen, alpha carbon, carbonyl. So this strand in the very center is going from top to bottom. The strand next to it, the one to the left of it, is going from bottom to top. So these strands are going in opposite directions. That makes this anti-parallel. And in fact, there's 2 copies of the protein here. The one on the left, you notice, is not connected to the one on the right. And even between the 2 copies, they run anti-parallel to each other. Right, so the whole thing is a 8-stranded anti-parallel beta sheet made from 2 copies of the same protein. right? So what's holding this together?
Same for Helix.
Right, same thing, hydrogen bonding between carbonyls and nitrogens. And if we try to get our distances between any two of them, we'll see it's, again, hydrogen bonding length. However, there is no formula this time, right? Because this strand could run off and do who knows what before it comes back and pairs with the strand it was just leading. So there's no formula like an alpha helix of n hydrogen bonding to n plus 4. No such mathematical relationship exists here. Okay. What if I were to take this strand, right, and you notice it has this twist to it. What if I were to take the ends and wrap them around such they would make a cylinder, right? Not let it just randomly go on left to right here, but grab the two ends. I need more strands than this, obviously, but if I had 11 or 13 or 17 strands or something like that, and if I take them and wrap the two ends such that they touched each other, I'd make a cylinder out of this, right? What would that look like? So let's close this and open up the 1JBY. Right? And again, this looks not very useful with all the R groups showing. Let's turn those off real quick. And can you see that as I turn it, I'm going to try to line it up so I can turn it easier. As I turn it, you'll see those beta strands are passing in front of you and then behind. If I put it at an angle, you can see that as well. They formed this cylinder. So this is called a beta barrel, right? And it may look like the cylinder is open on the ends, but it's really not. We've eliminated all the R groups right now, so there's many atoms you can't see. If I turn all those back on, you'll see all that. That there is no opening to this can, right? It's a molecular tin can. There's no 10 involved, but it's a can. Right? And what it does is protecting something in the center. Can you see in the very center of this thing that there is sort of a broken alpha helix? Right? So you see the beta barrel going around, and in the center there's this broken alpha helix. If I zoom in on it, ignore the lines going by in the foreground and background. Just look at the center. You see an alpha helix in there. Right? And what's happened is it's been broken down here because these atoms have rearranged to make the chromophore. Right? So this part here doesn't look like a typical amino acid. There is a tyrosine on the end, yes, but this ring is an atypical arrangement. Right? So that ring right here is made from amino acids just prior to the tyrosine, right? And if you look in our list, the tyrosine over here is number, if I can find it, I think it's 66. 66. Right, so tyrosine 66 on our list, which is right here, is listed as not tyrosine, right? It's been rearranged because the one right before it, right, has been merged with it. Right, so we've done some chemistry in here to make this rearranged molecule. And all we've done is extended the conjugated bond network, the double bond network, to make this a chromophore. So this is green fluorescent protein, and that's why it fluoresces green, because of that modified amino acid setup. Why do we need the elaborate cage? Well, this molecule is not stable. If you introduce oxygen to it, it will oxidize and ruin the chromophore. But to create it, I needed a molecule of oxygen. So this cage is developed. Its whole goal is to allow one oxygen molecule in it when it folded, and no more. So most of them do not fold properly with the oxygen. Very few do. And those are degraded and we recycle the amino acids. But those that are made are now useful as green fluorescent protein. Okay. All right, let me show you a couple more and then we'll get to your exercise. Let's close this one. I want to open 3FVB. Okay. So this one looks rather complicated. It's showing all sorts of things again, but let's eliminate some of the clutter, right? So let's take off our R chains one more time. And you'll notice that this is made of a bunch of alpha helices. Right? It's not a stranded beta sheet here. These are alpha helix. Right here, many, many more of them, right? A whole bunch of alpha helices throughout here. And in the very center of this thing is something unusual. Right? It's not part of an amino acid chain. It looks like what we showed at the very beginning. It's a polyporphyrin ring, right? And in fact, the metal in the very center is an iron, right? So here's an iron atom held in this polyporphyrin ring, right? And of course, the protein itself is holding that whole contraption. Okay, so what this molecule does is it's a bacterial ferritin, right? And ferritin's used to concentrate or gather iron. So what these bacteria do, this is from a Brucella species, so it makes this little molecule, if you have a Brucella infection, and lots of other bacteria do this too, and they release this molecule into your body. And as it's going around the body, it sequesters iron, taking it from you. And some of them deliver that iron back to the bacteria. So the idea here is the bacteria need an iron source, and they're not getting it without this molecule. So they're stealing it from you. So if you can inhibit this thing in some way, it could be an antibiotic. Right? Problem is, you have molecules like this on your own, like hemoglobin, and you'd like to not inhibit that one binding oxygen. That would be bad. Okay? So this is called a 4-helix bundle on the left. These 4 helices form a bundle. You notice alpha helices are not straight. They have a slight bend to them. Right? And they cusp around each other, and they form these bundles. There's another 4-helix bundle on the right. And this pair forms a great way to bind things like this heme group in the center. Okay. All right, one more to go. We want to open 1 BCF. Open 1 BCF. Right, and this looks very cluttered. Right, let's get rid of all the side chains. See what it looks like. So, does this kind of look like like half of a baseball? Right, if you take the typical baseball, its 2 outer flaps are sewn together along the seams. So if you imagine another copy of this thing sitting where this one is not sitting, it would form a sphere. Right? So this is kind of one flap of a 2-flap baseball. Right? And this is actually tobacco mosaic virus. Right? So many, many viruses use proteins as coats on the outside. And inside of it, you have some things that sequester iron, like the one we just saw. You know, this section here looks just like the thing we just saw. Right? So this thing is held together, and you notice there would be some inner space if you had another flap to make this into a sphere. And in the inner space would be where the DNA and RNA in this case would be held. Right? So that's how our typical proteins are put together in the viral coats. Okay. All right, lastly, we want to open 1 BQ9. Close this, open 1 BQ9. Right, it's a rather small protein. Okay, and if you notice in the very center, there's a high abundance of rings. Right, you've got tyrosine here, tryptophan there, phenylalanine here, another tryptophan over here. Lots of ring structures in the center. So the very core of this thing is very hydrophobic. There's also some leucines sticking their tails in there, right? And then on the very outside of this protein, you notice there's a lot of hydrophilic things, like these glutamates and aspartates, right? All sticking on the outside. Here's a serine sitting over here. So charged on the outside or polar on the outside, mostly hydrophobic on the inside, typical globular soluble protein. So this one is a very high resolution. We say it's 1.2 angstroms. Okay, so I want you to load the map on top of this, the 1BQ9 OMAP. You remember how to do that. We loaded the structure first, the PDB file first. Then we go to Open Electron Density Map, DN6. On a PC, tell it to look for all files. Then go to 1BQ9.omap and open it. Same parameters as before, and it should draw it on the map. And if I go click on one of those tryptophans in the center, which we're fond of doing, and center on it, like there, you notice that it's, again, shrink wrapped onto the tryptophan, but now it's even tighter. you can actually see a hole in the 5-membered ring, right? Because this is a much higher resolution map, meaning I have a better idea of exactly where the atoms are. The lower this number, the better the resolution. Okay, so if I have a 1.6 angstrom resolution, it's not as good as 1.2, which is not as good as 1.1, right? If you get down below 1 angstrom, it kind of gets hard to see because the cage network shrinks so much that it's hard to tell what things are. So this is an ideal resolution for looking at amino acids. If you had something like a 2 or 3 angstrom resolution, you wouldn't be able to see individual atoms because the resolution is longer than the bonds themselves, right? So you want something smaller than 1.5, say, to see individual atoms. And this is an excellent one. Okay? So what we're going to do with this one is try to assign some amino acids, right? So they're already done for you in this structure, of course, but it's again practice. If you pick one, like this proline that you mentioned over here, you'll just like proline. So let's zoom in on the proline and ask yourself, center, is it really proline? Or does it fit the density well? Remember what fit the density well means. Every line is inside of the cage, and every cage has a line in it. There's no empty cage network, right? Well, as you were asking me in the other one, as somebody pointed out, you see all these spheres out here with no lines in them. What are those things? Right? And in particular, if I find one probably by that serine, what are all these spheres out here? So this is a serine, and you notice there's a bunch of spheres around it. There usually are around serines. What are all these isolated spheres? It's clearly not part of the protein. They're not connected to anything. They look like single atoms. Anybody have a guess? There's actually 4 or 5 of them here. So what would appear as a single atom and be within hydrogen bonding distance of this serine's oxygen? It's kind of a tricky question. Right? Those are actually water molecules. Right? And you're thinking, hey, a water molecule is not a single atom. I agree. But in electron density structures, you can't see hydrogens. So water would look like a single sphere for an oxygen. Now sometimes you may see the little mouse ears where the hydrogens are often located, but most times they're tumbling so it looks like a sphere. Okay, so those are water molecules. So don't let them confuse you if you see a bunch of spheres without density in them. If they're not connected to anything else, they're most likely water molecules. Okay, so here's what you're going to do. And I'll open up, this is your assignment for later that I want you to turn in at the end of the semester. We're going to open a different assignment. We're not going to use the 2021. We're going to use the one I've used for the past like 15 years. And I'll do some of them with you. I'm going to save this one for your grade. So let's close this. Right? I want you to open assignment 3500. So open PDB file, assignment 3500, which should be in your folder. And as you look at it, you should notice there's a lot of one type of amino acid in the structure. And your assignment 2020 will look the same. It'll just be a different protein. So there's one particular amino acid that shows up abundantly in here. And you can look at the list and figure out what it is. Right? There's a ton of alanines in here. Do you think it's likely this protein is that many alanines? Of course not. I mutated them all to alanine. I know it's a cruel thing to do. But I want you to figure out what they really are, right? In order to do that, I need more information. I need some information of what the atoms actually might look like. So we're going to open the O map on top of it. So open the electron density map, DN6, change it to all files on PCs, go down to assignment 3500. You're going to do the assignment 2020 later for a grade, and open it, put it on top of it, and now I have information. let's say we'll do number 25 together. I'm picking one at random. So over here, instead of trying to find number 25, I can simply go over to this list and left click on 25 to highlight it, and then right click will center it for me, as if I use the centering function. So if I were somewhere else, if I click on 25 and right click, it centers it for me. I can zoom in. Looking at the R group, which I'm pointing to the left. I see the R group looks like an alanine's methyl group right now. Pointing to the left. Does alanine fit the density well? Or does it look like there's something else there? Right, if you want to clean up your image, you can remove all the other side groups and everything and just leave that one. And it might make it easier to see. Now all I see is that one amino acid. Right? It looks like there's something bigger. Right? If it were alanine, this sphere right here would just continue around as a sphere, and there wouldn't be any of this other density over here. Right? So it clearly looks like there's something else, maybe a big ring out here. Let's try putting something out there that would fit. How about a phenylalanine? Let's try that. So I click Mutate, click here, go to phenylalanine, click Mutate again. That's what I intended to do. Click Yes. Does it fit now? Well, I can't see the whole phenylalanine. You notice it cuts off at this edge. I told it to only draw it as a cube from 5 angstroms from my center. So I need to move my view out a little more. So I can move my center over here now. Now does it look like phenylalanine fits? Or should I change it to something else? Well, in this case, the end of the ring looks like there's something else out here. So like before, this isn't phenylalanine, it's most likely a tyrosine. So let's mutate it to tyrosine. Now does it look like it fits? Almost, if I could move this ring over a little. So I'll do that to convince myself. Now does it look like the tyrosine fits? I actually got it pretty well. Right, so I'm convinced this is tyrosine. So I can go back and turn on some other amino acids around it. You can leave them off if you want. I'm just turning them back on so you can see. I've done that one. I've done that. Number 25 is definitely tyrosine. It was not alanine. So I got to go through and do every alanine that's listed as alanine, because I know there may or may not be alanine. If you find one in the structure, like number 33 here, for instance, it says arginine 33. That one is definitely arginine. When I was creating the assignment for you, I deemed some of them too difficult for you to do, most likely because they're on the extremities and the density is not very well defined. So if you see one that doesn't say alanine, leave it alone. It's correct. If it says alanine on any of them, you need to check it. It's probably not alanine. There may be one that is, but it's probably not. And so I want you to go through and do those, each one. When you're done, here's how you save your files and how you send them to me. And this is the last thing we'll do together. Okay, so go to File. Let's say I've done all of these and I've assigned them how I want. I go to File, Save the current layer. Right? It's going to create a PDB file. Right? Call it something. Right? It doesn't matter what you call it. Preferably put your last name there or something so I know who it is. I'm just going to call it something. And then I save it. And what happens if I have yet to finish all of them? I'm going through, I just did number 25. I want to do number 26 and that happens. The program crashes. Well, I have to start over. I open the program, but I don't want to load the PDB file assignment 2020 or 3500 because I've already accomplished some things on it. I go to something and load that, and it's right where I left off. Number 25 is still listed as tyrosine. But I still want to load the exact same electron density map as before, because that's the raw data, that's not changing. Right? You'll never change your density map. You'll never save on top of it. But you can save your work, and you notice my tyrosine I assigned is still there. So along the way, it'd be a good idea to save every few of them, or every one. And you can also send this file to each other, and you go, hey, I think number 27 or number 28 is serine. I don't know what it is. and you send it to one of your friends or classmates, and they go, no, I think it's threonine. You missed the methyl group on it. Oh, I see what you're talking about. So that conversations of what I want to happen. All right, so looking at that. You can also save one more thing. When you're finally done, we're going to save the sequence. So we already saved the current layer. We want to save the sequence. We're going to call it whatever. I prefer you call it your last name. All right. And if you go in that folder and open up whatever, it just gives me the list of amino acids, right? So you see we changed this number 25 here to tyrosine. There's the Y. And what I want you to do is this won't say something. This will have your last name there, because that's what you called the file. I want you to grab all of this, including your name, including the caret, right, the greater than sign, right? Copy that, paste it in an email to me, and that's how you turn in this assignment. Once you've assigned all of them correctly. Okay, so the one I want you to do, and I'll stop and answer questions after this. So I'm going to close this one. The one I want you to do is assignment 2020. There it is. Right, it's not very long. It has 64 amino acids in it. So I want you to assign all 64. Some of them are done for you, so you don't have to do 64 of them, but many of them are still listed as alanine. Okay, so I want you to go into the thing, right? Observe the polyalanine backbone. Many of these are falsely assigned. I want you to go in and change them all correctly. Right, so when you're done changing them all, you're going to go to File, save your sequence. When you're done, call it your last name. I'm just going to call it whatever. And when I open whatever, it'll have, you want to change assignment 2020 to your last name, of course. And then you're going to send me this in an email. Right, just copy that, paste it in an email. There's a bunch of A's in here that are not A's. Right, you need to fix those. If it's a non-A already, leave it alone, because it's correct. Right, like number 8 is lysine, and number 18 is arginine. Right, but number 11, 12, 13, those that say alanine may or may not be. So go through and change those. And I'll work on some of these together with you. And if you want to watch the video again to learn how to do it, just go through and do that.